genetic modification, gene editing, CRISPR-Cas9, DNA splicing. No matter what you call it, altering the makeup of the most basic piece of life carries with it many different aspects, including pros, cons, and implications for the future. Before we begin to discuss the intricacies of genetic modification, we first need to understand just what genetic modification is. The first technical use of it dates all the way back to ancient times when our ancestors were able to selectively breed dogs. However, because this falls more in line with artificial selection and Darwin's principles, the first real use of the extremely advanced technology we know of today came in 1973. According to Gabriel Rangel from Harvard University's Science in the News, scientists Herbert Boyer and Stanley Cohen worked together to engineer the first successful genetically engineered organism. Their method included a very precise technique that allowed them to cut out a gene from one bacteria and insert it into another bacterium. The most popular technique used today for gene editing is called CRISPR-Cas9. The enzyme Cas9 is used as a pair of molecular scissors that cut two strands of DNA at a precise location so that specific bits can be added or removed. This works because the cell immediately begins repairing the cut DNA, and what results from the repairs could be different than what was there before. And because cells in the body possess a highly accurate and almost error-free DNA repair system, this is what allows the repairs to actually work when we cut the DNA out. Alternatively, it is possible for scientists to straight up place a different sequence of bases into cut DNA. However, it is a much more complicated process, and the repair process is preferred. This massive breakthrough opened up countless new opportunities for the science community to investigate this new topic more. Although, with such an exciting new technology on their hands, the science community decided it was best that they hold a meeting to design guidelines for genetic modification. At the Asilomar Conference in February of 1975 that took place in Pacific Grove, California, those in attendance decided on multiple measures with regard to gene editing. As written by Paul Berg from Nature.com, participants agreed on the final day of the conference that research should continue, but under stringent restrictions. The recommendations formed the basis of the official U.S. guidelines on research involving recombinant DNA issued in July of 1976. While these regulations provided structure that was needed in the early days of genetic modification, with how much the technology has progressed, it is possible that things need to be revamped, but not necessarily be made stricter. The best part of the new technology and how far it has progressed is that the sky is the limit. But that is also its greatest weakness, that it may someday be what ultimately wipes out all of humanity, which is why we have to ask ourselves the question, how far is too far? The possibility that humans may someday use genetic modification for the purpose of solving one of the world's most difficult problems is promising. The possibility that we may create humans capable of outperforming unmodified humans in every category is terrifying. However, these possibilities are things that we have to live with if we wish to let this technology reach its full potential. With adequate funding and research opportunities, we may be able to solve the ever-growing problem of world hunger. With the ability to harvest more meat and food from genetically modifying animals and plants, we could feed every mouth on the planet in no time at all. Not to mention the implications with disease that gene editing could have. The greatest field that would be greatly impacted is the cancer field of disease. Because cancer relies on rapidly producing cells, if we could snip out the piece of DNA that codes for cell reproduction in cancer cells, we could effectively stop it in its tracks. Not to mention the possibility of other genetic diseases that are formed in the fetal stage of human development and things that we could snip out of DNA and possibly change in developing humans. Therefore, when we ask ourselves how far is too far, we must take into consideration how far we could go and how far we could progress as a species if this technology is allowed to reach its full potential.